Hello everyone. It is a great honor to be here today to introduce our study. Microbial ecological memory of recurrent drought enhances host stress tolerance through enrichment of plant beneficial bacteria. This study addresses a central question, whether recurrent drought, as a typical climate extreme, can generate a form of ecological memory in plant-associated microbial systems. Using the desiccation-tolerant moss Calohypnum plumiforme, which toggles between dry and wet states, we simulated 4 natural drought cycles. Our results show that recurrent drought not only reshapes microbial communities but also generates a stable ecological memory. This memory is mainly localized in the green, photosynthetically active phyllosphere, accompanied by continuous enrichment of plant beneficial bacteria and functional reprogramming of the microbiome. Overall, our findings support a holobiont-scale ecological memory linking plants and their associated microbiota. The highlights of this study are summarized in 3 aspects. 1. Niche-specific memory. Ecological memory manifests via deterministic assembly in metabolically active green parts, while senescent tissues and soil remain largely unresponsive. 2. Beneficial bacteria as carriers. Methylobacterium and Sphingomonas are the cornerstone of this memory. Their enrichment and functional gene upregulation directly enhance host stress tolerance. 3. Holobiont framework. The convergence of microbial functional restructuring and host metabolic reprogramming substantiates the holobiont ecological memory framework. Our first key question addressed whether microbial communities can form a stable ecological memory under recurrent drought, and in which niches this memory occurs. Our analysis revealed that, first, regarding overall community structure, the green tissues exhibited the most pronounced shifts across the 4 desiccation rewetting cycles, whereas senescent tissues and bulk soil showed relatively muted responses. This indicates that microbial responses are not uniform but exhibit clear niche dependency. Further analysis demonstrated that as the drought cycles progressed, the microbial community in the green parts displayed 3 key characteristics. 1. The community structure gradually stabilized. 2. Community predictability increased, indicating an enhancement of deterministic assembly processes. 3. Beta diversity declined, suggesting that the community converged rather than diverged. Concurrently, a pivotal finding was the sustained enrichment of plant beneficial bacteria in the green tissues, with Methylobacterium and Sphingomonas being the most prominent. Therefore, our primary conclusion is that ecological memory does exist, but it is niche-specific, occurring primarily in the green tissues and characterized by the enrichment of plant beneficial bacteria and stabilization of the microbial community. Our second major finding further elucidates how ecological memory forms. Metagenomics revealed significant functional restructuring by the 4th drought cycle, characterized by enhanced symbiosis genes, stronger microbe-host ties, upregulated signal transduction, enhanced cell-cell communication, increased Kz and carb metabolism, improved resource use, enriched elemental cycling, more active ecosystem function. Concurrently, the host metabolome shifted, marked by fluctuations in flavonoids, indoles, and organic acids. Structural equation modeling revealed a closed-loop coupling of community dynamics, functional restructuring, and host metabolism. Conclusion: ecological memory is not merely microbial, it is a systemic holobiont memory forged via the coupling of microbes, genes, and host. Finally, the core conclusions of this study can be summarized as follows. First, recurrent drought induces a distinct ecological memory phenomenon within the moss phyllosphere microbiome, characterized by strong niche specificity. Second, this memory is not merely a shift at the community level but constitutes an ecological process driven by the convergence of enhanced community stability, the sustained enrichment of plant beneficial bacteria, and the coordinated restructuring of microbial functions and host metabolism. Building on this, we propose the holobiont ecological memory framework, positing that recurrent environmental stress integrates the microbial legacy into the host stress memory system through bidirectional microbe-host interactions. This study extends the research scale of ecological memory from soil systems to plant-associated microbiomes. Future studies involving the reinoculation of keystone taxa are warranted to further validate their direct causal contributions to host drought tolerance.